just a question, it's human nature, a question of trust. If you suddenly gave me all the questions from Urbana, would I give them to you as practice questions? And if I gave them all our questions up here, would they use them as practice? And would you guys who went to Illinois sit there and share them with your compatriots who were down there? Sure. So it has been a process over the summer of actually coming to understand each other and actually creating different pools of questions, some which are for formative things where you test yourself, and some that are for summative exams, ones in which we use to rate whether or not you've learned something, okay? But over this term, then, the final goal is to actually have a complete sharing of those pools. Now, that said, each year we generate a whole bunch of new questions, all right? And so, and some questions get stale, and some have 99% of you get it right. And that might be because something is very important and everybody should get it right, or it might be that everybody knows that every fraternity file has that in, they always asked X. And we can distinguish those because we can always change the wording of a question, and then all of a sudden it goes back down to 70% hit rate, So it is something that's in flux, and the idea, though, is that what is being presented to each of the classes at each location is the same. And this becomes more complicated, as you imagine, in the second year, not the first year, where the ways that the different sites have organized themselves are different. Some do it basically by organ system, and some don't. So that's why it's in a state of flux. If you talk to the folks who are your SIBs in M2, for example, the only difference in the way that we're going to examine you this year is that you no longer have, microscope slides and microscopes to contend with. Everything's online, okay? So what were microscope questions will be image-based questions on the electronic testing. Yeah? Are they not going to tell you you want to see the question and answer? Sure. You'll be able to know which, let's suppose that you blew off my lecture today and just came in, right, and didn't know what you were supposed to be looking at. It will reveal which lecture it was that you failed at, okay? It will tell you, there's an association both for chapters in the book and for the lectures of what things you did well on and what things you did poorly. So you'll know that. You'll know a given number of questions. Let's say you got all 100% of those questions right on cardiovascular, but nothing right on molecular motors. All right? That will come back to you. You will not probably get the questions with the answers.
What's the harm in that?
Well, we actually ran the experiment 2 years ago, and it turned out to be very few people actually came to look at the exams. In general, looking at exams works when the problem is essentially one of problem solving. In other words, how can I improve my ability to solve problems? And then if we go through the questions and answers. Since most of the tests here, unlike cell and tissue biology, aren't problem solving. Here, you're saying, can I learn to recognize something? So that's more or less like it. The test had been run using biochemistry, and they were just answers, facts on this thing. And they started out with 50 people in the room, and it went down. By the end, they had fewer than half a dozen. And the final one, they had 2 people show up. So it was just people voted with their feet. And with that, I'll give it over to the next lecture.
I think it's fair to say, though, that what I've told past classes is that how do you study for an exam? Well, you start with lecture. And what is posted on Blackboard is an extension of the lecture, or what's called note aids. Another thing you consider then is what is in the lab manual book. And actually, those are the primary sources there. And then of course, you're responsible for what you're pointed at in the textbook. It's unreasonable for us to require everything that's in the textbook. It's too much. There's no textbook that's just right. Ross is a good textbook. In fact, it's used by about 80% of medical schools in the country, in North America. There are about, 127 medical schools now. And the Ross text is the dominant use, because it's about just right. And that's why it's used a lot. So that's how I would approach it. lecture plus handouts, lab manual, and use the textbook as a reference. Now, of course, the best case would be if you had a chance to look at the paragraph headings in the chapters before you come to class. So you get a better understanding what the main areas that are going to be presented, and maybe make a hierarchy in your own mind of how you take notes. Now, my particular note aids are pretty much tabula rasa. You know, I'll show you pictures, and I fully expect that you'll be handy working with your fingers taking notes. You know, that's important. That's input. You know, there's such a thing as a proprioceptive sense. You not only see what you're writing when you take notes, and you maybe hear what's going on in the room, but you feel it. You feel it. That's an important part of note-taking. So anyway, I've given you in my handouts, you can just write all over them, and do it that way. Now, regarding the lecture of the hour, fortunately, the chapters in the textbook look very close to the ones that Mike Ross used from me years ago. So I could do some song and dance up here and maybe show you a few things you don't see in the textbook. But as a fallback position, the text is really good on blood cells and a blood cell formation. Really good. Of course, the slides you'll be looking at virtually in the if it's a lab manual, is something else, something completely different, as Monty Python used to say. And that is not duplicated in very many places around the country. So, by the way, you know, you can ask us questions of that sort at any time. It's just usually you probably shouldn't use up too much of this class time. Otherwise, because like I say, I'm going to turn into a pumpkin in a few minutes. Now, what happens if you get cut off? Well, like I said, fortunately, I'm not going to give you much that you can't find already in the textbook. It's not possible, by the way, to be comprehensive about any topic you get from any discipline in the College of Medicine. Too much. The curriculum was designed for the state of our knowledge in 1920, when you could cover basic sciences in 2 years. Now, you couldn't cover basic sciences in 6 years. In fact, as I confidently estimate, all of our knowledge in the basic sciences turns over twice in your medical school lifetime. Think about that. That has tremendous consequences for when you get out into your clerkships and your residencies. I know of residencies at a very famous institution south of here who had to deal with different protocols practically every month for certain leukemias. It just changes that rapidly. All right. Let's, blood. Now, we're not talking about blood, the fluid here. Blood's a really interesting soup of proteins and other materials. Major protein is albumin. There's some fibrinogen that is the fibrin clotting origin protein. And that's about 7% protein soup in the blood. But what we're really talking about are a cell component of blood. And remember, these cells we're going to be talking about are the endpoints of development. In the second half of this hour or so, we'll be talking about making those endpoints in places called bone marrow. But we're going to be talking about the endpoints. And just clip off the tops of our knowledge on how these look in preparations called blood smears, and in a few cases how they look in sections, and under the electron microscope in very thin sections. I think a good way to start actually is to, I found a way, by the way, by using this podium I can make that arrow bigger, which seems to be what should be done. Let's talk about these formalisms here. Let's get this out of the way. Peripheral blood cell parameters. Well, you can divide the blood cell world into two colors, if you wish, red and white. Now, blood is red when the red cells are oxygenated, but it turns brown very rapidly when they're deoxygenated. But we're going to talk about red because we're also going to be talking about the cells after they're put down on a glass slide and stained with dyes. And the particular dyes are a dye called eosin, E-O-S-I-N, and it is a dye that carries a negative charge at the pH of water. And another dye from a family called the Romanowsky stains. Principle is azures, which are an azure-colored dye, which carries a light positive charge at the pH of water. And so anyway, dividing that world into red and white, or red staining with eosin and not so red staining with eosin and white blood cells. Let's talk about RBCs first. If you look at a slide, and Dr. Art showed you one a few days ago, basically what you see is a lot of pink. It's like a pink smear. And it's because in making a smear, you want to make a monolayer. But we know from some hemocytometer studies, the unit we want to talk about is how many cells per cubic millimeter. And there's about 4 or 5 million red cells per cubic millimeter. Now, at the outset, as an aside again, I want to say, let's deal with this unit, the cubic millimeter. Now, textbooks, they do vary just like the flavors of the authors that write them. For some reason, this edition of Ross, they want to tell you how many cells there are per liter of blood. You know, so you get numbers like 10 to the 9 and 10 to the 12 and say, no. Let's do what's done generally in the classic and historically in medical schools and deal with a number per cubic millimeter. So there's a few million. These cells are regularly about 7 to 8 micrometers. Some years I like to say they're 7 micrometers. Other years I like to say they're 8. It's all right. You know, none of them are perfectly that anyway. There's a range of sizes. But they are that regular, such that they're intrinsic scale bars. So if you see in your virtual microscope a suspect cell that you suspect is a red cell and say, yeah, it's a red cell, you know that that's a scale bar in that view. That is, let's say, 8 micrometers. And everything else you can scale based on that size. Nuclear shape, well, Na, not applicable. The circulating red cell does not have a nucleus in mammals. It does have in development. We'll talk about that later. But there are some, like amphibians and snakes, reptiles, birds, do have red cells that retain their nuclei. They're nucleated in circulation. They don't have any conspicuous granules, either azurophilic, that is azure-loving, or a so-called specific class. And they have a pretty long lifespan, 120 days. This really gets to be interestingly important the past few years when it used to be if you're a diabetic, you would get your blood glucose measured. Well, now what is done is they gather or assess the percent of a certain subtype of hemoglobin called hemoglobin A, in particular A1C. The A1C subtype of hemoglobin has a hemoglobin that gets glycosylated. It binds glucose. And it lasts 120 days. So it's a wonderful tool for telling what your blood glucose content was for the past 120 days, not just some stick point in time. It's wonderful. Very wonderful. And that number, the magic number, it's about 7%. Less than 7%, at 7% you're marginally diabetic. Less than that, you're normal. And above that, you get to be diabetic, and more so. Let's look at the next column, the platelet. The platelet is smaller than red cells. They're about 2 to 5 micrometers in diameter. Now, the red blood cell is a biconcave disc or donut. But the platelet is a biconvex disc in the circulation. It's a fragment of cytoplasm. It has no nucleus. There are very important organelles in it that are responsible for its motility and ability to aggregate and do its work in counteracting hemorrhages. No cytoplasmic, well, it does have cytoplasmic granules, but they're so small you only see them by EM. And the lifespan is on the order of 7 to 12 days. Now these numbers don't memorize these numbers. Memorize sort of, their order of magnitude and rough numbers. So the platelets at the top there is about 1.4 times 4 times 10 to the 5 per cubic millimeters of blood. A lot less than RBCs. Now, if those are non-white blood cells, what about white blood cells? Well, they're white because the cytoplasm doesn't stain profoundly with eosin, so they're not very red. And the total white blood cells are much, much less than the number of erythrocytes per cubic millimeter. Notice down here, normal total, 8 to 10,000 per cubic millimeter normally. In leukemias, they can get much higher, much, much higher. Now, for the rest of these, we're going to be talking about the percentage of this 8 to 10,000. And the most numerous one is in this first column to the right, about 20 to 25%, not the most numerous, sorry. 20 to 25%, second most numerous, is the lymphocyte. It's regularly about the size of a red cell, as we will see. It has one nucleus. It might have a little dent in it, where in life, will be a centriole. And they live on the order of days or months. Monocytes, 48% of white blood cell total. They're much bigger. They have sort of the same body plan, as we'll see, as a lymphocyte, but they're bigger. In fact, relative to the size of a red cell, if we take that to scale bar, the lymphocyte is a cell that has a nucleus that fills most of the cytoplasm, and about the diameter of a red cell, whereas a monocyte is about twice the diameter of a red cell. It has a nucleus that occupies most of the cytoplasm, and it's mononuclear. It has a few granules that are azurophilic. Now, when you talk about white blood cells, azurophilic granules were named by their dye propensities, binding propensities, long before we ever knew what they really were. These are, in fact, lysosomes. What is a lysosome? That's a problematic term, historically, because it had a very broad umbrella term very early on. A lysosome was a membrane-bound granule, or loose slush, that had hydrolases active at an acid pH. That is the basic definition of a lysosome. Now, trouble is, there's a lot of membrane-bound organelles in a cell that have that property. They have hydrolases that are active at an acid pH. Not all of them look like granules or primary lysosomes, unused lysosomes. A lot of them have a mixed content because that granule lysosome is fused with another membrane organelle to make a bigger body that has a heterogeneous content. But you could still call it a lysosome. So if an unused primary lysosome, anything downstream might be called secondary lysosomes. Secondary lysosomes. I just want to mention that because I think one of my role is so you can be acquainted with what's in the literature, all the textbooks that are written to this point. How do those terms get to be what they are? What do they really mean? How many of them are archaic, and how many are just downright misleading? All right, monocytes are short-lived. They differentiate rapidly, produce rapidly in the bone marrow. They exit the bone marrow by the blood system. They actually dive right into the blood vessels, and they're taken off, and then they end up in the peripheral tissues as cells called macrophages. What does macrophage mean? Well, the word phage means from the Greek to eat. To eat. And macro means big eat, big eater, big eater. Meaning that these cells can gobble up a lot of extraneous content and come back for more. They're not postmitotic. They can make more lysosomes. They can chew them up more. They can spit out the residual bodies more. They keep going throughout life. They're not endpoint cells. They're not postmitotic. Interesting, the next column, the neutrophil, was once called a microphage. It was an eater. It was a one-time eater. It ate bacteria, and because it was postmitotic in the tissues, the nucleus was tied up, it was heterochromatic, completely inactive. It could not make more lysosomes, could not make, to put into granules, could not spit them out and generate more cytoplasmic content. They die. They are like cruise missiles in the blood that go out in the tissues, find bacteria based on a sensation, a chemotactic gradient, engulf them and house them and digest them. But then they die. That's it. They're macrophages, one-time eaters. Now, when you have a major infection, a lot of bacteria, a lot of neutrophils will come out of the circulating blood, will kill a lot of bacteria, and that creamy little material that you see in wounds called pus are the carcasses of neutrophils. And that pus will be cleaned up by guess what? Some macrophages will come around and it will chew up and digest that pus, and such that it'll be dried up eventually and something you don't need to know about now. Eventually a scar is formed from that expelled material. So a monocyte is a pre-tissue macrophage that circulates, and neutrophils are microphages. Now, neutrophils are the most numerous. In a smear of blood, about 60 to 70% of the non-red cells you come across will be neutrophils. They will be slightly bigger than the diameter of a red cell. Their nuclei have lobes. That is to say, they're not one big, simple, open nucleus like in a monocyte. The nucleus, as I said, is postmitotic. It's closed down so you can see visible lobes. And we'll see all this, what that looks like. You can have 2 to 5. A 5-lobe neutrophil is older than a 2-lobe. There are situations where you have neutrophils that might have many more lobes. That's kind of a pathological situation. But normally in the blood, 2 to 5 lobes of closed-down nucleus. It has 2 kinds of granules. The urophil, they have lysosomes. Sure they do. But they also have, now we get into another term for the remaining 3 columns there, the neutrophils, the eosinophils, and basophils, the so-called specific granules. Now I'm going to give you a take-home thing to do. It's the specific granules that characterizes really the function of these leukocytes. The specific granules in neutrophils, for example, are fairly small, about the size of eosinophils. They dominate the eosinophils. And they're very faintly stained with pink, and they'll show you all this. And so the cytoplasm looks like it's filled with a very pink, stippled little background. And specific granules contain antibactericidal proteins and bacteriostatic proteins. Sometimes these are called defenses. They have a few other things too. But think about that to start out with in your matrix. If you have neutrophil, eosinophil, basophil up here, and down the side you have specific, what are the specific granules? Neutrophils, like I said, they're like cruise missiles. They come out of the blood based on a need to kill bacteria. They circulate peripherally in hours or days. Let's go then to the eosinophil. Not very many, about 1 to 4% of that 10,000 white blood cell count per cubic millimeter. Their diameter of a neutrophil. Their nucleus regularly has 2 lobes, and I mean regularly. Regularly. It has granules. There's just a few eosinophilic granules that are lysosomes. It's the specific granules that caused the early histologists to call them eosinophils, because their granules are large, Even with a light microscope, you can see they're large and distinct, and they're highly eosinophilic. That is, they bind eosin in a big way. They bind eosinophilic in a big way because the specific granules have a protein called major basic protein. It's a protein that has a plus charge. It's going to bind the eosin, which is a negative charge. A simple salt kind of reaction, plus binds to minus. And again, they circulate on need hours and days. Or I'm sorry, hours and days for neutrophils. About the same, 8 to 12 days. Again, you're going to find different numbers on this. The point is they don't circulate 120 days, and they don't circulate 2 minutes. They're on the order of days. Now, basophils. Basophils are the least common of white blood cells. 0.5% or so. In other words, so if you look at 200 white blood cells, you'll see a basophil about one out of every 200. There's a diameter of the neutrophils, the eosinophils, and its nuclear shape is sort of 2 to 3 lobes, but it's sort of in an S shape. It's not really distinct lobes like the others. And its granules are just profoundly azurophilic because they contain heparin sulfate, which is a compound that is highly negative, and it's going to bind azure, which has positive. So their specific granules give it a very basic azurophilic color, and the specific granules. So they have azurophils to be sure, but the dominant azurophilic granules are the specific granules in the basophil. And basophils are poorly studied because it's hard to get enough of them and well enough to study. It's not clear. The function of the basophil is very closely akin to a cell you find normally in loose connective tissue called a mast cell. And they do not come from the same precursor. They come from different stem origins, but they seem to have like a convergent evolution of function. And they have a lot of this similar contents in their granules. Now, we'll talk about ways of classifying these in the next slide. There are a certain number of circulating, slightly mature red cells called reticulocytes. They contain a few remnants of cell organelles. We'll look at those. And in the course of making a red cell, you end up having to shut down and throw away all the organelles except for membrane proteins and contained hemoglobin. You spit out the nucleus. But a few are retained earlier on until they're fully mature. They're competent to carry gases. That's fine. But they have this remnant called a reticulum, which is from the Latin net or reti. I made a note down here for 3 lysosomes, and basophils have azurophilic granules, and their specific granules are stained with azure. Now, very importantly, and this is something that is the basis for a lot of NIH research these days, is circulating so-called stem cells. The concept of the stem cell is that if you take a seed, put it in the ground, you make the first little shoot as a stem. And it grows and it puts out branches. And it grows and puts out branches that have branches. And so forth and so on until you get a tree. All generated from a single stem. Interestingly, the term stem cell was coined, as I'll show you in a panel, by a fellow named Alexander Maximov. He was a Russian who escaped Russia just in time to be able to escape, and ended up at the University of Chicago around 1917. And he took his knowledge. He was a profoundly important intellect in terms of histology. And he started writing a textbook at the University of Chicago. That was joined by another Chicago department, an anatomist named Bloom, who finished the textbook. That textbook became Maximoff first, histology, and then Maximoff and Bloom. And then later on, around the 1940s and 50s, the head of the anatomy department at Harvard, Don Fawcett, joined William Bloom, and then the textbook was named Bloom and Fawcett. And finally, the later editions of that, after Bloom died, the textbook was called Fawcett, and there's still textbooks of Fawcett around. That textbook was the medical student histology book for about 70 years. 70 years. That's the one I used when I went down there. I have a PhD from USC, and I took the whole medical first year. And in a sense, I learned my histology back there. So, in a sense, I can tell you can have confidence to my component input into this course, because I know. I mean, in the way of teaching histology, the knowledge of histology, so-called Chicago-Harvard axis, I know what that histology looks like, and it was on that basis really that we picked the Ross textbook a long time ago. So you're going to learn mainline histology here, and you can be confident in it. All right. I talked about this briefly. Erythrocytes. Did you know that nitric oxide can be carried by hemoglobin? It is. It's not carried very far. It's a short-term, hormone. It, of course, is called the molecule of the year a few years ago. Now, maybe interesting for you to recognize that it is a major active ingredient in drugs known as Cialis and Viagra and such. It's due to the activity of nitric oxide. Platelets, and again, this is the cliff notes of cliff notes, blood cell functions. The platelets are anti-hemorrhagic agents. They're small biconvex discs. They bounce around on the inside of blood vessels. If there's a hole there, like in a hemorrhage, the platelet will bump into the connective tissue that's just outside, and it will bind. It'll stick. And so what you have are platelets. First of all, they will just stick to the hole, and their bodies will kind of fill it up. And then they'll be turned on, and they will secrete, and we'll show you in a panel, catalyze the formation of fibrin from fibrinogen. So they make a plug physically. They send out tendrils when they're activated. They sort of try to pull the walls of the hole together. They secrete in concert with the endothelium enzymes that cause fibrin clots. And there's one-stop wonderful little guys, because after the blood flow is plugged up and the endothelium, that is the wall of the blood vessel, is repaired, they pull back the edges and they retract the clot, and they end up just dissolving themselves and eventually taken up by macrophages. So they're very wonderful, and since they're easy to get to, a lot is known about platelet function. Neutrophils, I said, they're antibacterial agents. Basophils, by virtue of their specific granule content, one of the most important specific granule content are heparins and heparan that cause blood vessels to come apart at junctions. And the result is that you get some blood outflow between endothelial cells. You also get dilation of the venules to promote the activity of leukocytes getting out into a tissue where there might be a bacterial infection or inflammation. Eosinophils you might think of as anti-basophils because they have compounds that neutralize the heparin, that neutralize the histamine, and they have histaminase. Look at these what's in these specific granules? They're very easy questions to ask on exams. And it's important to know because that's what gives them their functions. What is in these specific granules? Monocytes are source of tissue macrophages. Lymphocytes are anti-foreign material agents. You'll have a lecture on the histology and superficial cytology of lymphocytes. And there, of course, there are a couple different types, the so-called B and so-called T. And there's also a kind called a natural killer cell. And we'll talk about that briefly, but you'll hear a lot more about that in your lecture on lymphoid tissues. Now, how can you, over the years, there are several ways to think about classifying these types. You can classify them based on whether they have a nucleus. Well, clearly red blood cells and platelets don't. You can classify them based on whether they're motile or not. I mean, in themselves, are they capable of, let's say you're talking about a barge channel. Now, a barge isn't necessarily motile in itself. It can turn its engines off and it can just float down. So yeah, red cells and platelets can float down with the flow. But in themselves, they cannot walk on the bank of the canal. Neutrophils, basophils, eosinophils, monocytes, and lymphocytes have motility. That is to say, they have motility by forming focal adhesion spots and then moving cytoplasm forward and making more focal adhesion spots and then moving along. And it was a kind of amoeboid kind of movement. So they're inherently motile. You can talk about, on the far right, whether they have granules or not. Platelets, well, like I say, there are a few teeny granules that will show you. Neutrophils, basophils, eosinophils have granules. Monocytes, and it's usually, I have to back up and say, historically, in your textbooks, you're going to hear the word, see the words granulocytes as opposed to non-granulocytes. Well, that was based on a light microscope. Monocytes have granules and some lymphocytes have granules. You couldn't see them with a light microscope. So they're classified as non-granulocytes. Well, so the formal granulocytes in the hematological literature still remain neutrophils, basophils, and eosinophils. But understand, the monocytes have eosinophilic granules and lysosomes. And that lymphocytes even have some large granules in them. In particular, the kinds of lymphocytes that have those are the kind that are implicated in being natural killer cells. And I'll show you that also. Now, what about making a smear? Smears are made still all over the world because they're fast. And a fast way of taking a snapshot of the blood cell population of whatever patient you were dealing with. All you do is just take a drop of blood and drop it onto a slide and try to make a thin layer of those cells. And you let it dry, then you stain it with the stains of azure and eosin, and you get red cells that are pink and white cells that have the granules that I'm going to be showing you in different colors. Now, blood is whole blood. It contains cells and plasma and white blood cells. If you spin down blood, red blood cells are more dense. They'll end up in the bottom, and this number of this fraction of this tube is about 40%. Sometimes it's called the hematocrit. And a normal percentage of the blood that you find in one of these tubes, it said 3 to 50%, but usually it's about 40% you want to be in a normal percentage of red blood cells that are in that pellet. White cells don't pellet so densely and rapidly, and so you find a thin layer here, so-called buffy coat. That's where the white cells are. And suspended, no reds or white cells are in the rest of this straw-colored fluid. That's plasma. Now, in terms of terminology, you might hear and see the word serum a lot. Well, serum is plasma that's coagulated to get rid of all the fibrinogen. And that is serum. So anti-sera, really, early on, before we knew much about how to make specific antibodies, antibodies were raised and sera were taken out. Because that's about as purified as we could get. And so you'd had antibodies against something or other in an anti-serum. So plasma, you coagulate out the fibrin, you end up with serum. Here then again is what the process. You hit a drop of blood, you take the edge of another slide and make a drag it across this direction or that direction. And what you want is to get a monolayer. So you can make easy counts. You don't want it to all be bunched up. And here's what you see, as you can just see here. There's 1, 2, 3 non-red blood cell contents and all these many other entities. Most of them are red blood cells. Now, pointed with arrows are little dots that are at higher magnification. You can't tell this by looking at this magnification. But I'm just going to tell you, these are platelets. Some of them are aggregates of platelets. But these is what platelets look like in a quick and dirty smear. Now, smears are not very good for cytology. You can't tell much about the cytology. You can not tell any ultrastructure from a smear. The main attribute and the benefit of a smear is that it's fast. You know, you can get one of these smears done in minutes. That's why it's still one of those things that are used around the world, no matter what culture or what country you're talking about. Now, in a prep specifically made, this just kind of drives me crazy that I have to generalize this all the time. So I have to keep this moving in order to keep the arrow, apparently. Let's see, or can I do that down here differently? Let's see, no. Automatic arrow. Let me keep it constant. How about that? Now, this is what you would want a smear to look like. You'd want to be able to see these red cells in the scanning electron microscope view. The scanning electron microscope provides you topographical views. A fancy word for saying it shows you the shape of things. You can look at it, turn it around in your hand, you can look at it. You can see that these are concave donuts without holes. You see these frilly cells. These are smaller ones. You see, they're about the diameter. This is 10 micrometers here. These are about the diameter of red cells, so these must be lymphocytes. Don't know what this is. It's some non-lymphocyte. It could be a monocyte, perhaps. It could be some granulocyte. You can't tell by looking at the surface of it, not with a scanning EM prep. But the point is, you can see these are kind of beautiful. Now, just to illustrate how deleterious a smear is, this is what it looks like. And these are red blood cells prepared just by air drying like you do in a smear. I call this my breakfast slide. The force of surface tension is pulled through these cells about 100,000 pounds per square inch. So here you still get a lot of hemoglobin here, and it doesn't rupture, but you lose the thin membrane on the red cell donuts here. This used to be one of those white cells. It's a pancake. And down here we have sausages, which are platelets, a single platelet here and an aggregate of platelets here. Smears are fast. They are not great in terms of cytology. So let's rip through these real quickly. Here's your red cells, about 7 to 8 micrometers in diameter. A few platelets out here. A non-red cell. It's big. And just by the size of it, you see this is a nucleus stained with azure. And the cytoplasm is kind of pink. And it's not a lymphocyte because it's too big. You don't see big granules in the cytoplasm, so you know it's a monocyte. We'll get to more of that later. And of course, in sections, red cells, you'll see them in blood vessels. Again, there's scale bars. So you can, this is a fat cell out here. So you can see, all right, this is 8 micrometers. You might say, all right, this fat cell is 1, 2, 4, 5, 6, 7 times the fat diameter of a red cell, so it's about 60 micrometers there. You saw this before, scanning EM views of aggregated red cells. In the TEM, transmission electron micrograph section, in the plane of that torus, this is what a red cell would look like. If you cut it across, it would look like this dumbbell-shaped form here. Here's a little platelet down here. Notice it does have a few granules in it, but not big ones. And notice how small it is. A couple interesting things about hemoglobin. One is that it was called the first molecular disease, coined by H. Stanley Bennett and Linus Pauling, going back on an airplane in 1948. Linus Pauling was working on DNA, and what was discovered was that if the beta chain of globin in hemoglobin, if you had a single substitution of an amino acid, a hydrophobic amino acid for a hydrophilic amino acid, that is, say, a valine for a glutamic acid, that hemoglobin would become sludgy in a lesser aerobic environment. So that hemoglobin normally confers on red cells, it's very slippery. It can slide through blood vessels, the diameter of which, inside diameter, is less than 8 micrometers. It can be 4 micrometer diameter inside, and the red cell will still slip through it. But not hemoglobin with sickled, HbS in it, as it's called. That substitution of one amino acid caused this problem, and that was due to a single base pair change in the codon. I mean, that's remarkable. A single base pair can cause this disease. Single base pair can cause a difference. And what is it about sickle cells? Well, they look sickled like this, and they're very rough and tough. They're not flexible. Some clinicians sometimes talk about people that have sickle cell disease as having rocks in their kidneys or rocks in their heads, because these tend to get hung up in small blood vessels, like you have in your cerebral cortex or in your kidney. In fact, here's an example of the TEM, the picture. You don't expect to know this is kidney. I'm just going to tell you. Here's a capillary kidney, and here's a cut. Look at this. This hemoglobin doesn't look nice like the hemoglobin we saw in a TEM. It is rocky, and it gets sludgy in environments like in some places where you don't have as much oxygen in the capillary circulation. Now, if there's a silver lining to having sickle cell anemia, is that you probably won't get malaria. Turns out that malaria, are a parasite that eat out the inside of red cells for hemoglobin. Here is a SEM view of the developing a parasite, and the hemoglobin has been all eaten out. In cross-section, it looks like this. This is that parasite, and the cortex is still there from the hemoglobin, and the cortex of the red cell actually prevents this organism from being recognized by the immune system. Pretty insidious little bugger, this guy. And this sometimes is called a falx or a flap, and hence the term P. falciparum, falciparum, Plasmodium falciparum. And here's a light microscopic view. Turns out the malaria don't like HbS. And so the chances are that you can be somewhat resistant to malaria if you're unfortunate enough to have sickle cell anemia or sickle cell trait. Let's talk about platelets. In the circulation, here's a platelet in circulation. It's a small disc. Here's the edge of a blood vessel here, also down here. Here's the outside world of cells. There's the outside world of cells. Here's the inside world. We're looking down on the lining of this blood vessel, down and on in here. We're seeing some red cells here, some red cell here, here, and some lymphocytes probably, because of their size. And So normally in circulation, it's a little biconvex disk. By EM, this is from your textbook. Have a look at it. We're not going to dwell on the, like I said, there's a lot of plateletology out there. I want to deal only with the superficial functions of platelets. I don't really much care about L granules versus alpha granules and delta granules and that kind of thing. This is what they do. Platelet thromboplastin catalyzes prothrombin into thrombin. This is initially soluble, but it becomes eventually a fibrin polymer clot. And then the platelet also has platelet plasminogen activator. On the other half of this arm, that catalyzes plasminogen to plasma, which dissolves the clot. So the platelet makes the clot, or participates in making the clot, and also participates in dissolving the clot. Pretty neat. Pretty neat. An activated placement is not biconvex disc anymore. It sends out little tendrils. And these are what activated platelets look like. Notice the scale here. This is 1 micrometer. So this platelet is sending out tendrils. There's actin in these. And this facilitates their aggregating and sticking together to make that plug. Let's go to white blood cells. Here's a granulocyte. This is the first one, the most common, so-called neutrophil. Notice it has 2 to 5 lobes. It's hard to say. You know, this nucleus is definitely not a eukaryotic nucleus anymore. But let's say there's one here. Don't know. Maybe it could be 2 right here. One here and one here and one here. And then you have a little one right here. This little one is interesting because it is a so-called Barr body, B-A-R-R, which is an unused X chromosome in the female. And females mostly have neutrophils that have Barr bodies. And these was once used, I don't know, I don't know how the latest data was used, but it has been used in past Olympics to determine whether what sex you were, whether you really were male or whether you were a female. Because it turns out there were some rather large women from Soviet Union, shot putters, that were breaking world records, and it turns out that I remember one woman's name was Tamara Press, very cause célèbre there. And it turned out to be a guy. So now they actually have better tests for that gender business. But nevertheless, the Barr bodies is still there. This is a little lobe. It's called a Barr body, unused X chromosome in a female. All right, here's from their textbook. Here's an EM view. Looks like there's maybe 1, 2, 3 lobes, 4 lobes here. A lot of granules. There's Golgi in here. And here we have, again, a neutrophil. It's not completely segmented here. It has a lot of small granules in the cytoplasm, but it's sort of pinkish. Neutrophils, I said, are bactericidal entities. They have on their surface receptors, this again from your textbook, for binding either by virtue of binding to directly to antigens or by using a complement receptor or an FC receptor and an antibody. And they take these up and they will then dump the Azurafil granule lysosomes into this phagosome. This is a body that has taken up something or other that is eating something body, a phagosome. And when the lysosomes bind to it, the jargon historically is now you can call this a phagolysosome. You can also call this a secondary lysosome if you want. So if the primary lysosomes are whatever color we're doing that with, let's see, I guess we're doing blue here. And this is now a secondary lysosome, whereas the blue are actually primary lysosome. They digest, and sometimes they spit out the content. By SEM, here is a neutrophil taking up a yeast particle. Make a little chalice-like flowing over. 1 or 2 taken up here. Eventually, they'll close up here and do the digestion. Basophil looks like this. Notice the specific granules here. They're very purplish and dark and intense, and much so that you can't even see the nucleus below it. Big purple granules, specific granules of a basophil. The EM of that from your textbook. Big granules stained, their specific granules stained with the azure dye. Very profoundly. You'll be able to see all these in your lab manual exercise also. The eosinophils. Now notice, one very regularly, one lobe here, one lobe there. 2 lobes. What you want to do is focus through and verify the other 2 lobes. If you have big red granules now, big granules, not the little ones in a neutrophil, it's stained profoundly with eosinophils. And from your textbook. One thing of interest in eosinophil is useful for identifying EM pictures on exams, for example, is that the eosinophil granules have a crystalloid on the inside. And it is, in fact, this crystalloid that's the major basic protein. The content of the granule, the non-crystalloid content, has a lysosomal content and other proteins that are used to kill infectious worms, helminthic worms, and other parasites. Now, here's a cell, a white cell. This does not obviously have granules in it. It's the size of a red cell. See here. Nucleus takes up most of the cytoplasm. There's a little thin rim of cytoplasm out here. This little small lymphocyte. Looks like this. Sometimes you see a dent. This is where centrioles would be typically. Here's a nucleus here. Here's another small lymphocyte here. Small lymphocytes of the B variety will become activated and they will enlarge and either multiply or they will go on to produce what are called plasma cells in the connective tissue. Plasma cells. Plasma cells are specific antibody-producing, super antibody-producing cells. All the antibody produced by a single plasma cell is so-called monoclonal. They all have the antibody against the same epitope on whatever antigen it was working against. Yeah?
Can you go back one slide and just one more time clarify what 4 steps is right now, the next 1 and 4? I just didn't catch.
This one here?
No, all 4. I just didn't catch, like, inactive, activated.
Oh, this is small lymphocyte that we looked at before. Most of the nucleus occupies most of the cytoplasm. Okay. When it becomes activated, you get into a proliferating form. The so-called medium-sized lymphocyte. That's this one. So you see, it gets more cytoplasm. It gets more organelles in it. Okay. Now this is a proliferating form. It's also the form that differentiates further. Some of which will differentiate further into along the line of making a plasma cell. So if it goes along that line, you start making endoplasmic reticulum. And eventually, you make a lot of it, and the cell looks like this. And eventually, it will burst and deposit that antibody into the connective tissue. Here we have a cell that is a lymphocyte, but notice it has granules in it. And I just want to mention this, just to show that even with a light microscope now, you can say that lymphocytes can be called granulocytes. Here is a neutrophil out here. Notice cytoplasm. Granules are small, they're not large. Here's another view of this so-called this lymphocyte that's now sometimes called a LGL, large granule lymphocyte. There's a large granule, large granule, large granule, large granule, large granule, big nucleus. And it's from this population that seemed to be, we can define the natural killer cells. Finally, the monocytes, they're big. Twice the diameter of a red cell. Same body plan as a lymphocyte, but they're big. They have granules, but they're not profound in the light microscope. And here's a summary. Neutrophils here and here. Eosinophils here and here. Basophil here. Here's actually a circulating plasma cell. Sometimes that happens. Not many. It's a trace. Lymphocyte here small, lymphocyte here small. It's a disruptive process is making a smear, and this is just actually a nucleus that's been pushed out of a lymphocyte, and it's a destructive situation. So this is not anything you want to identify it as anything. It's a nucleus, former nucleus of a lymphocyte probably. And here's monocytes, here, here, here. They have a big indented nucleus. Sometimes I call them a Pac-Man nucleus. Remember Pac-Man a long time ago? A big indentation. Yeah.
If you go back a few slides, I want to ask you a question.
Which one?
You said like there's a lymphocyte and there's a neutrophil. I do if they're the same size. Yeah, these two. How do we distinguish these two? You said like one lymphocyte because of the lobes in there.
This is on the small side for a neutrophil. I wouldn't use this in a question. It's on the smaller side. Usually the granulocytes are about a red cell and a half in diameter.
We shouldn't be able to know that that's a lymphocyte and that's a neutrophil.
Oh, yeah. Look at the nucleus. Lobed. Yeah. Just that on itself.
Can you repeat the question? I couldn't hear what you asked.
He wanted to know if this is a neutrophil or not. And all you need to see here is the lobe of the nucleus. You never see lobes like this in a lymphocyte of this side. I wouldn't use this as a nice example of a neutrophil because it's too small. They're usually bigger than that. So we're talking about ranges, of course. All right, we better move on to the next. Oh, you can figure that out. This is easy. What is it? What are these? Right, what are these? Look at two lobes, big granules. What's this? Basophil. What's this? Yeah, what's this? Yeah, you pass. Yeah, we got to go on next. Well, I don't want that. I want... Come on. All right, we got to move along, otherwise we're going to run out of time. Now we're going to talk a bit about blood cell formation, in particular the tissue that most of the blood cells start out in, at least. Here's that summary slide again. Now these cells in development, all blood cells, start in the yolk sac. Now, and I'm going to have to do this again, otherwise I lose my arrow. Okay. Started on the yolk sac in the first trimester. Second trimester is dominated by presence of developing of red cells and white cells in the liver and spleen. But by the third trimester and then on into perinatal life and adult life, cells are formed in bone marrow. What do I mean by bone marrow? We mean you haven't had bone, you haven't had marrow, now you're going to have marrow. We're talking about long bones, for example. These cells are formed in the insides of long bones, and in the course of development, you do get this kind of an approach. This is a tear away from the bone. There's a bone here, you don't know that officially. But it's very cellular, very, very cellular. It's just a zoo of cells. And we don't ask you to be able to identify the individual stages. In the course of life, the cellularity in long bones will decrease. And it will actually move to other places, the vertebral columns rather than the long bones, vertebral columns, for example, iliac crest. And it still has the capability in long bones, but it looks more like this. It's mostly become fatty. A lot of these little white cells are fat cells. There still is a capability. There's a lot of stem cells in here for the capability of making blood cells. But even in bone of your age, a lot of long bones are going to start looking like this. And a lot of the cellularity formation of blood cells is moved to your vertebrae, ribs, iliac crest, sternum, such places as that. Now, if you cut across a bone, you have here, I want you to know that the inside is highly vascularized. From the outside, from a nutrient artery, and also across the bone through conversion systems, such that you have a very rich vascular compartment. And those blood vessels inside of bone marrow are called sinusoids. They are actually post-capillary in position. Now, you don't know what post-capillary means. I just want to say they're really on the venular side of the circulation. Here we have bone down here. There's not much I want you to recognize, except later on, once you have blood vessels, you'll be able to recognize this. These are the sinusoids I want you to pay attention to here. Real thin-walled structures. Most of these cells are about the same size, except for these big, large cells down here. They're called megakaryocytes, meganuclear cells. So, blood vessels, blood cells developing outside of blood vessels, and megakaryocytes developing outside. In mammals, blood cells are formed outside of blood vessels, outside, extravascular hematocytopoiesis in mammals. In some other organisms, you actually can get blood formation inside of blood vessels, but not in you and not in mammals. By SEM, again, these are these so-called sinusoid vessels. That doesn't mean they're sinusoid-shaped or cosine-shaped or something like that. That's just a developmental word. And outside are all these developing blood cells in this scanning EM view. If you take a bone marrow and dry it and crack it open. Here again is a lumen of a sinusoid. All this zoo of developing cells out here. And I just want you to note this very thin little lining cell here. Here it is again. Look how thin. This is not a formidable barrier to these cells out here. The reason I say that is because there needs to be a concerted activity between the cells out here that are developing to maturity and that cell, to go across. It's more of a chemical barrier than a physical barrier. This is more like a lock and key that the cells out here need to go across into the circulation when they are mature. The concept of the stem cell. Let's see if I have this. This is the guy Maximoff I talked about. At a congress in Berlin, he first postulated the existence of hematopoietic stem cells in 1908. He brought this idea to the United States and the University of Chicago. And he developed the theory, so-called monophyletic theory of blood cell formation. There were others in the field that thought that the red cells had their own line of stem cells, that all the others had their own line of stem cells, but he was the one that championed the idea that there were a possible single cell. So you have a stem cell tree that's a multipotential for both the marrow and the lymphoid cell lines. And from that splits off a pluripotential hematopoietic stem cell, only the myeloid cell tree. And then you have a committed stem cell, for example, for neutrophils. Incidentally, operationally, the stem cell is operationally called a colony-forming unit. And that's operationally defined as if you put a cell in a tissue culture dish, whether you can cause that to form that population. Here is one person's view of this stem cell tree. Here you have a stem cell down here, a very primitive cell. In marrow, the marrow hematopoietic stem cell. There's also a stem cell in marrow that's a marrow mesenchyme cell. These may be the same cell. We don't know. There's a lot of argument about the relationship of all these at this level. Interestingly, the The umbilical cord blood is a very rich source of stem cells. It used to be that when a child was born, umbilical cords were thrown away. And now they're sold because they're a very rich source of stem cells. And these stem cells that are in cord blood are isolated and are very, very effective in bone marrow transplantation. There's some of the best takes and most high proportion of takes in marrow transplantation include cord blood stem cells. You start off with this. The first thing that splits off is a progenitor for T lymphocytes, and then a progenitor for the myeloid B lymphocytes. B lymphocyte progenitor splits off. The myeloid progenitor comes up here, splits off. The erythrocyte megakaryocyte goes up here. You get the granulocyte monocyte. This goes to the monocyte chain. You can read all through this yourself. I say osteoclasts not because at one time it was thought that osteoclasts that are important cells in bone marrow are derived from circulating monocytes. That's not so. The certain histiocytes and macrophages in liver cells are from monocytes, but osteoclasts are not, and the microglia in the nervous system are not. Osteoclasts rather are produced by an earlier progenitor off down here, and they rely on activation by some interleukins on this osteoblast stromal cell progenitor. So, are these the same? There's a marrow mesenchymal cell and a marrow hematopoietic stem cell, and are they the same? Well, turns out an entrepreneur at Stanford University found a way to isolate stem cells by subtraction. And without going into it very deeply, he was able to separate out all the other cells, and he found a cell that actually had no lineage on it, at least at this time. This is old news now. I mean, this is not even up to date, but it never makes the point. Let's say it has, for our purposes here, only CD34 plus and no other lineage. And the progression requires interaction with other marrow stem cells. Upon induction to progress, it proliferates to form colonies. And now these are called colony-forming units. Question is, what does this look like? Well, a cell is about the diameter of 7 to 10 micrometers, round shape, nucleus round with indentations, nucleoli 1 or 2, large, chromopatterned, finely dispersed small aggregates, cytoplasm, no Golgi apparatus observed, no ER, no lysosomes, lots of free ribosomes, a few mitochondria, and not many organelles. So what does this look like? If you had, which does this look like most? Does it look like an erythrocyte? Does it look like a neutrophil? Does it look like a lymphocyte? Does it look like a monocyte? Actually, it looks most like a large lymphocyte. In fact, there is a population, long known, there's a population circulating called large lymphocytes. Here's a small lymphocyte here. See this? Nucleus occupies a lot of the cytoplasm, but it's very condensed. This is a memory cell, it's not doing much. It's surveying all the tissues as it goes through the bloodstream. But this cell is about twice size. It has a big eukaryotic nucleus, euchromatic nucleus rather. Not much heterochromatin. Here's a big nucleolus. A large lymphocyte. That's what this looks like. Now, it turns out in the body, it's hard to study these in tissues. You study this. Almost all you know about this is from culture. But it seems like they tend to hang up in the sinusoids, the sinusoid blood vessels in liver sinusoids, and that does make some sort of sense because the liver does have a capability to make blood cells upon demand. If you lost all the other places to make blood cells, liver could do it again. So could spleen. In this drawing, this stem cell might look like this. And from this becomes all the lines. So if you want to get to this point here, this erythrocyte, how do you morph from that to get to this? It's pretty self-evident here. This is from your textbook. First of all, you have to make ribosomes. And with the stains we got, that means the cytoplasm turns bluish because of the azure stain in the ribosomes. Eventually, you get hemoglobin being synthesized, and these have these names. They're polychromatic because they have varying amounts of pink and blue that are grayish. Then what you have is a nucleus starting to fold up, and the size gets smaller. Eventually, the nucleus is gone, and then you have the erythrocyte. Here is what a cell looks like in the red cell chain just before it's spitting out the nucleus off to the side. These, again, is what are reticulocytes. Here is a red cell with no organelles remaining. Here's one that has some organelles remaining. And it's sort of a reticulum, or net, a little network. These are, remember, about 0.5% of circulating red cells. And here is what one of those reticulocytes sort of looks like. A few mitochondria, a few ribosomes. This can carry oxygen and nitric oxide and CO2 perfectly well. But eventually, in order to be in the circulation, it has to get out of the bone marrow. This is bone marrow over here. This is the lumen of a sinusoidal blood vessel here and here. Here's a cell here, this little thin, little fragile cell that's the barrier. And here's a junction between these two cells, here and here and here and here, here. Now, this cell does not go through junctions. Remember, it's not motile. It has to work in cooperation with this endothelial cell, and it forms by a little pore, a transient pore, and this will come out and move in concert with the endothelium. It's not exactly clear how this works, but they don't bust up the junctions. They use selectivity in the lock and key approach to unlock a door, a transient door, to get into the circulation. Here we are. We're looking down on one of the sinusoidal blood vessels. Here's a margin between the 3 endothelial cells. Notice, coming into the lumen at you is this little probe from a red cell. Once again, bone marrow. Let's now look at these big cells. In 1906, a guy named Wright, who developed a stain, nice stain for platelets and for blood cells, made these drawings where these megakaryocytes pushed processes into the lumen. And he drew these long processes in certain other places in the body, and it showed that the platelets were derived from processes from megakaryocytes. This caused some guffaws in all his presentations he made. Remember, we were talking about 1906 or something like that. They were made fun of him because he was doing tales about how megakaryocytes were defecating into the lumen of sinusoids. Turns out the guy was right. It took only about 70 years before scanning electron microscope. Here you see one of those sinusoids here, margin here, margin here. And notice this process coming across the wall into the lumen. Here's some lymphocytes here, there's a red cell here. What's going on there? Here's another one of these in the circulation. Turns out you can strip out the cytoplasm off of one of these megakaryocytes and make about 20 of these long processes. So it's real. And eventually, in the 1980s or so, they got around to studying this in culture, and indeed it was. You got these processes produced before you segmented into platelets. There's an error in your textbook that shows that on this topic is a diagram. Remember, diagrams can err. The body doesn't err, but somebody who draws a body can make an error. And in that drawing, it shows megakaryocytes kind of fragmenting off platelets and them going individually. That's not how it works. They go by making processes first. All right. Well, anyway, you can walk through this because I guess we're going to shut off now. I can keep on talking if you wish. Here we have. Well, maybe I can short circuit that by saying we're not going to ask you questions on any of this. How does that do it? No, I want you to know how to morph, though. What do you have to do to get a red cell? Come on. What do you have to do to get a red cell out of this? You have to throw away nucleus, you have to get smaller, you have to make ribosomes to make hemoglobin. Understand what you have to do to get from here and there. Same way here. What do you have to do to get from this myeloblast to a cell that looks like this, or like this, or like this? Well, it turns out what happens in life is, first of all, the cell gets much bigger. And this stage is called a promyelocyte. And the nucleus gets bigger. And the first things you see are azurophil granules. Maybe all it's ever going to have. That's one stage. The next identifiable stage, and there's all kinds of stages in between, but benchmark stages. The next stage is the nucleus kind of flattens, and you start to see the specific granules dominate. So in the basophil myelocytes, the specific granules are purplish. Here they're pinkish in the eosinophilic, and here they're small pinkish in the neutrophilic. Then the nucleus folds down. At this point right here, mitosis stops. So this form here, all this over here are postmitotic. So here the nucleus is going to fold down to it looks like this. Here this nucleus will start becoming two lobes. Remember, it started out like this. Comes down to two lobes, and it doesn't progress any longer, any more so in an eosinophil, two lobes. Down here, you get the lobes formed, but you do get a form that looks like this. And again, and this one shows, this drawing shows 1, 2, 3. It's 2 to 5 is typical. So again, what do you have to do to go from this stem cell type? Hematologically, all these are really called blast forms, B-L-A-S-T forms. And blast is one of those words that's used in a lot of contexts. It means forming. It's a forming cell. So, for example, a fibroblast is a fiber-forming cell. A chondroblast is a cartilage-forming cell. An osteoblast is a bone-forming cell. But anyway, in the context of blood cells, this would be called a blast. I want you to know how you get here. You're not going to get asked by me anyway any questions that specifically. I want you to identify a neutrophilic metamyelocyte, although it's not too difficult. Just think, well, you can tell this from this, can't you? And you can tell that from that. And certainly you know the endpoints. So in these ranges here, it can be difficult. Therefore, if it's that difficult, it's not. Pardon me? They start with the same blast, yeah. And then what happens is first of all, the monocytes differentiate so rapidly that except in culture, you probably can't distinguish any forms with a microscope. See, because they still have the big nucleus in a monocyte, maybe they pick up a few lysosomes, but they look pretty much like a blast cell. And they hang around outside the sinusoids, and then they move in on demand. Same with the lymphocyte. The lymphocyte, you're going to get downsizing and making cells that look like large or medium-sized lymphocytes. And then in bone marrow, what happens is so-called B cells are produced in bone marrow throughout life. In fact, bone marrow is the biggest former of B cells, much bigger than the lymphoid tissues. But the progenitor cell, like we took in the stem cell tree, at the progenitor level, the progenitors for T cells will move to the thymus out of the bone marrow. But yeah, to get to a progenitor B cell, a progenitor T cell, just need histologically, and that's what we're talking about, is the downsize it. You got to get smaller. That's all, just smaller. And yeah, this is a study in culture, and many studies are dealing with this kind of thing. But what you can discern in the marrow of a light microscope is pretty difficult, if not impossible. Now, one thing about the process of making granulocyte from myeloblast to, say, mature, and here we're talking about neutrophils. Another word for, probably more used in the clinical literature for neutrophil is PMN. P stands for poly. M stands for morphonuclear. Polymorphonuclear means many formed nucleus in cell. Polymorphonuclear cell. In other words, it has lobes. You can have 2 to 5 lobes. That's polymorphonuclear. As they're called PMNs, or sometimes they're just called polys. So neutrophils commonly are called polys, commonly they're called PMNs. But this we're talking here about polys now. they have the same progression whether it's a eosinophil or a basophil. You have the myoblasts we just talked about, the promyelocyte, and the myelocyte. These all can divide and proliferate along their lines. Then you have the postmitotic or the metamyelin. Notice densing of the nucleus. Now we're getting a C-form nucleus, and now we're getting 2 to 5 lobes here. And this takes 6.5 days. And in blood, the neutrophils still hang outside the sinusoid. And when they're in the blood, their half-life is pretty short. And then in the tissues, again, a couple days. If you were to take a snapshot of the population of bone marrow and characterize the developmental populations, and this is a snapshot, most of the time, look, 60%, 20% here, 20% meta, 20% bands, 20% polys. In other words, 60% aren't even in the circulating. They're in the maturated storage pool. At any given time, maybe 15% are the myelocyte stage, 10% of the promyelocyte stage, and 5% in the blast stage. So this adds up to 90%. So of the totally white blood cells, only about 10% are actually in the blood. And that's what I say it's really important to consider that all these are hanging outside here, waiting for a stimulus to come into the circulation. And they get in like the red cells do. Here's the lumen, like an L up here. And here is that endothelial cell. You'll learn to know and love that this is called a simple squamous epithelium. You'll learn about epithelium right away tomorrow. One of the types is a single cell layered, very flat cell, thin cell, called squamous. Simple squamous epithelium. Well, that's what lines the blood vessels generally. So here's the lumen, here's the endothelium. And notice it makes a little pore here, in concert here. It sticks a little finger of cytoplasm. This here now look, this lobe here, this is almost certainly a poly, PMN, neutrophil. Sampling out here, and it will, again, in concert with this cell, will form an aperture. And that aperture will close. How do we know that? Well, I just showed you. Well, maybe it's in the next slide. Let's see. Look. Does this look like Swiss cheese? No. This has had a lot of those cells coming through like this. And here is the perspective. We're sitting in the blood. We're looking down on the floor, which is the surface of the simple squamous endothelial cell and epithelial cell. Same thing. Here is the margin of one goes out over here. Here's the junctional margin of another. You have this coming through. Now, when you consider how many of these has gone through into the blood here, if it was not temporary apertures, this would look like Swiss cheese, and it doesn't.